Welcome, everyone. Today we're diving into the world of data and focusing on AI and the challenges that big data poses for AI.
And the human hunger for data means the more data we feed into an algorithm, the better the AI should perform, at least in theory.
In theory. Now, this can sometimes lead to issues, such as the problem of averages.
Exploring big data, AI, and the intriguing problem of averages, that is what matters today on Discovery Matters.
Let's get cracking. To walk us through big data and the power of artificial intelligence, let's say hello to Matej Macak.
I'm a partner from McKinsey. I have a PhD in machine learning and neuroscience. I used to run experiments on neurons and to figure out how much they can learn and how much information they can store. And then I would also run computational experiments to try to summarize all of those insights that I found in the computer. Essentially learned both the biology parts as well as the machine learning components, which now I'm using these components in helping my clients as part of the Quantum Lab, which is the machine learning arm of McKinsey, to accelerate drug discovery processes.
Matej began his career by looking at neurons. And do neurons and machines learn in the same way?
So this is a major area of contention in the field at the moment. There's a big debate around the similarities and differences between human neurons and the neural network neurons in models like ChatGPT 4.0 and other large language models.
Many of the architectural components that the machine learning specialists are still doing when they're building the neural networks, they're inspired by the real thing. However, in many respects, they're different. They're simpler, right? So there are many chemical components potentially and many complex processing that these neurons do not have. But at the same time, some researchers are saying that the simplicity is actually their strength. Some leading scientists in this area, like Geoff Hinton, have recently come up and even said that simplicity may be so powerful that actually make them even more suited to some of the tasks than the biological part. Like the neuroscientists and computational neuroscientists, they still work very, very closely in bringing the best learnings from like neuroscience into the computational science as well.
So does Matej mean that they're essentially building a network of problem-solving nodes?
Well, neurons are the units that integrate all of the information from your senses, hearing, vision, and so on, your proprioception, and they make a decision about the information being strong and important enough to send the signal further on. And that decision process is very similar in both biological and artificial neural networks.
Neurons are actually incredibly complex structures. There are many different types, with many different ways how they are shaped, what inputs they receive. They do not just make decisions about sending or not sending signals, but they make many gradual changes, like chemical and otherwise, that make them respond in a much more nuanced way than what the artificial neurons can do. And many of the artificial neurons are built in a way to maximize the potential of the hardware that we use to train them. Many of the decisions that we've made in computational science have been driven less by biology, but mostly about how do we squeeze most performance out of the available hardware. What then changes is the actual implementation, how to get there, right? Which is actually quite a fascinating and beautiful thing in some ways, right? Because you can learn from these differences and push the science of machine learning further.
So let's get down to the use of machine learning in drug discovery, protein design, and so on, because we've talked quite a lot about that, and we're really interested in that. AlphaFold has had a lot of coverage and a lot of interest in terms of understanding the shapes of proteins, for example.
Sure, and we're seeing a lot about the use of machine learning to generate candidates for drug discovery. So I'd love to start with the underlying question of why somebody who is working in drug discovery would use machine learning to identify potential drug candidates for therapy.
Some of the knowledge graphs that we currently have that cover the known knowledge about biological pathways and biological interactions, they can have tens or even hundreds of millions of nodes. So human would not be able to comprehend many of these components. And so what machine learning does is that it's a very, very powerful way to summarize the information. And then you can use that information to make predictions, such as which chemical binds to which protein that can be involved in a disease. Or you can make predictions about which proteins are likely on the biological pathway that leads to a disease. At the same time, there are many other use cases that people leverage, such as the ability of machine learning tools to help interpret images, text, and so on, that many researchers work with through microscopes, through imaging modalities like MRI, and so on.
What types of relationships are these machine learning models seeing across biological structures and biological functions?
The modern microscopes can often generate millions of images in a week, and there's no human who would be able to process this amount of information. Yet that information can be incredibly valuable, right? Because it can tell you whether there is a potential drug that has been applied in one of those microscopy images that could be a new treatment for patients. The second thing is that often these machine learning tools think a little bit differently than humans do, because they learn from the data that you provide to them. When we have been working with medicinal chemists and others, you would find that they are sometimes surprised by the suggestions that machine learning tools generate. But that's a good thing, right? Because that then often sparks a really helpful discussion that can progress the approach. The reason why these tools are capable of doing this is that they are often trained on millions of images or chemistries or potentially biological pathways. And it kind of gives you like a summary of a specific pathway that may be linked to a specific disease. And then you can use to make predictions, such as, is the specific drug going to be likely effective in discovering new treatment? And there have been many successes already in this space where the tools have been leveraged in this way.
We need some examples now.
I'll give you one. New antibiotics have been discovered using some of these tools.
We've seen situations in cell and gene therapy space where new drugs that are now in clinical trials have been discovered in 6 months rather than the usual 18 months. We were able to squeeze the amount of time that you would normally take in a manual and traditional discovery through this in silico-like method. So it is still early days for a lot of these tools. Clinical trials and drug discovery can take many years before the compounds actually make it into the market. But there has been a significant level of advances in this space, and the space continues to evolve.
But is it about generating more candidates or generating better candidates?
The main thing is accelerating the generation of quality candidates that get to the patient as soon as possible. So you can do that with more, or you can do it with better.
You want to have high probability of success. The likelihood that the drug actually is going to treat the disorder that you're trying to treat. If you are able to do many experiments in silico before you actually try them in real experiments, you're saving a lot of time, right? Traditional wet lab experiments, they can take many weeks, right, to run sometimes, and even months, right? And so if you are able to shorten this, it can be incredibly powerful. At the same time, as we mentioned, these tools, they think slightly differently. So they come up with different candidates, different approaches, right? There can be gems like hidden in those approaches that can have meaningfully different chemistry and activity that can have a high therapeutic potential. This increases the probability of success of these drugs in the real world.
These tools are faster, they have a higher likelihood of success. So what could the implications be for clinical trials, given that they generally take a long time and face numerous challenges?
Yeah, and that's the longest, phase in getting a drug to market. So to fully understand it, we've got to look at the broader challenges that the drug discovery community faces, not just speeding up the process and improving the likelihood of getting a drug that has a therapeutic effect. So we asked Matej, what are the other hurdles that need to be addressed?
These tools are new, and they often come up with potential suggestions that may not make sense, even though there is actually something very valuable in the suggestions that the machine learning tools generate. And so what you find out is that these tools, they work to enhance people. People need to know how to use them most effectively. They need to understand why these tools are making suggestions. The challenge is, how do you most effectively create this flywheel effect where the initial hypotheses generate better data, and that better data creates better models, and then that creates this really reinforcing virtuous cycle. So it is as much change management and process change problem as it is a data science and a machine learning problem.
It's interesting, isn't it? It's almost always the case that science isn't the hard bit, it's the people bit that's really difficult.
But as Matej said, these are the tools that can make people better.
It's what we call the cyborg model, right? It's the best thing when you combine the researchers with the insights that are coming from the machine learning models. Because one thing that we have found is that the experts, they often sit on tens of years of pattern recognition and experience of knowing this is probably going to work or this is maybe not going to work. Have you considered this kind of approach? And what we found out is that the tool can suggest initial ideas, and then the researchers, for lack of a better word, riff on that idea and then create a new and better solution. I think cyber model hopefully is the place where you have the machine learning and the expert knowledge coming together to make better medicines and therapies for patients.
That's fascinating. I love the idea of evolving what it means to be a problem-solving being. And of course, my personal experience with AI so far matches that. I mean, I'm not using AI to make medicines or generate drug candidates, but for me, it's about writing tighter and faster for a communicator like me. I mean, so far, it's pretty fun.
Yeah, it's fun, and it's like having a partner in crime, as it were, isn't it? And Matej himself, he has a real passion for this.
I definitely bore many of my friends with the excitement that I have for this topic. For me, it's really, at the end of this exercise, there are drugs that are going to help treat patients. That's what we're trying to do. And at the moment, it's really expensive and really hard to find new drugs. It costs about $2.6 billion to create a new drug. And if I remember correctly, about 10% of the drugs actually make it from the clinical trials successfully. This is a massive drop-off and massive challenge that the pharma industry and drug discovery process need to surmount. But if we find therapies for many disorders that are currently untreatable or difficult to treat, or people suffer from many side effects that make their quality of life low, the excitement of the fact that some ends there may be a drug that I was part of in helping to design is probably the thing that really drives me day in, day out. And it's a super exciting area to work in.
So using large data sets can be extremely helpful in predicting the efficacy of drug candidates in their early stages. But it isn't always just a benefit for treatment. It can lead, interestingly, to the problem of averages.
My name is Tom Yankeelov. I'm a professor of Biomedical Engineering, Diagnostic Medicine, and Oncology at the University of Texas at Austin, where I'm fortunate to direct our Center for Computational Oncology. However, the most interesting part about me is that I've married so far beyond my skill set, and I have a lovely wife and two wonderful children, and I've won the lottery 3 times.
Tom is using modeling to predict breast tumor response with digital twin models. He is avoiding the use of averages to tailor treatment more closely to individual patients.
What we do now is all based on population averages. Getting a new drug to FDA approval, at least in the States, is 10 years and $1 billion. So it's a very long, very costly process. And what you have at the end of the day is a therapeutic regimen, an intervention of some kind that works best on average for some average population. We just wrote a whole paper on this. I'm designing clinical trials for patients who are not average, because if you take everybody's height or weight, you do get a nice normal distribution. But once you start moving behind height and weight into more substantive characteristics, there's no normal distribution. It's not just that our moms told us everybody is different and special. It's really quantitatively true, right? So the notion of trying to come up with a therapeutic regimen that works for the average person, you're almost coming up with a therapeutic regimen that works best for no one, because there is no average. If you build a cockpit for the average pilot, you're building it for no one, because no one fits in that scheme. And that's just physical dimension.
So instead, Tom's using digital twins and taking measurements from the individual, and he uses this in the mechanism-based model that can make predictions about the actual individual's tumor.
How are digital twins helping us get away from this population average to individualized medicine?
There's 3 key components. Digital twins employ mathematical models to represent the physical object, predict its behavior, and then use that knowledge to optimize the intervention. And another key thing about digital twins is there has to be this interplay between the physical object and your model. In our case, in oncology, a patient will appear, they'll have some diagnostics, for example, of a solid tumor. We make a host of measurements. Those measurements are then used to calibrate a mathematical model. That mathematical model can then make a prediction. The goal is to use that prediction to guide the intervention, which then changes the system and the behavior of the tumor because there's been an intervention. So then you take more data and use that to then update your model and make a new prediction. And there's this back and forth. That's a key thing about a digital twin. We take a suite of semi-advanced MRI measurements. You don't have to have 20 PhDs hovering over the scanner to get it to work. So we take a series of measurements. There's the high-resolution anatomical data that sets the domain that we work on, the computational domain that lets us segment, in the case of breast cancer, the breast into adipose tissue and fibroglandular tissue and the tumor. And then we have data that tells us about blood flow, vessel permeability, tissue volume fractions. And then we have other data that tells us about cell density. And we use those to calibrate a mathematical model.
Once it's calibrated, you can then run it forward in time with the digital twin. And then hopefully that will tell you the best way to intervene.
Why magnetic resonance imaging specifically? Why not CT or ultrasound as an imaging modality?
MRI is a really wonderful technique because it's very flexible. You can make lots of different measurements in one scan session of a patient. You can get your high-resolution anatomical stuff, but you can also look at blood flow and vessel permeability and tissue volume fractions, cell density, which then over time could lead to looking at things like cell proliferation. You can look at hypoxia. There's a whole host of things you can get in one scan. By and large, MRI is an extraordinarily flexible technique. Ultrasound is a wonderful technique, but the contrast to noise ratio that you get in ultrasound is not as strong as in some of the other modalities. PET imaging is phenomenal. The FDG PET is the workhorse of clinical PET and tells you about glucose metabolism. So we are working very hard, actually actively right now, to extend our mathematical models to take that information, because of course, it's been known for over a century that tumors have abnormal metabolism, so you can see where the drugs get in the body.
So Tom has said multiple times that it's been highly accurate in predicting response of triple-negative breast cancer patients to standard chemotherapy.
So what does that actually mean for a patient?
If you had a way that could predict early in the course of therapy that this intervention was not working, then you would switch course. And unfortunately, we assess the response to therapy rather than predict it. In many cancers, there's this thing called this international standard called RECIST, the Response Evaluation Criteria in Solid Tumors. Patient's been diagnosed with a solid tumor malignancy, they get high-resolution CT, MRI, and they measure the longest dimension of the tumor. Then the patient goes on therapy, and they'll have a follow-up scan at some number of weeks or months down the road. But the key is that it's all assessing response. So if you have progressive disease, you've wasted time. You have the side effects of whatever the intervention was. In the case of triple-negative breast cancer, if you're predicting that the current intervention is not going to work, then you would say, okay, maybe we go to surgery sooner and spare the systemic therapy toxicities. Maybe there's a clinical trial that the patient can go on. Those kinds of things can be thought about.
Now, in an ideal world, the physician would be able to work with the patient to choose the right therapy to start with or intervene quickly if that therapy proved ineffective. And the aim is then to give doctors the actionable information within 24 hours of receiving that patient's data.
So what are the challenges then in getting this rapid turnaround time to be a real and robust recurring possibility?
There is precedent for this heart flow that does this for cardiovascular disease. They take CT images before and after contrast of the chest. It's sent off to the cloud. Their analysis does this to see whether or not a patient needs to have a stent placed optimally. It's a real success story in computational medicine. So that's how you have to navigate the barrier of getting the data out of the hospital, making sure the data is completely de-identified, so all the patient health information is secure, sent to a place where the analysis is done, and then sent back to the hospital in a way that the treating clinical team can handle it. So we want to do centrally located, and where you just have to build one server to do this, because it is computationally intensive, and they describe how the tumor's growing, how it's invading tissue, so how it's migrating, how it's proliferating, how it's responding to therapy.
But change in the medical profession takes a long time, and that's for good reasons. The medical profession is a small c conservative.
So what are physicians saying to Tom, and what does he anticipate coming down the road in terms of organizational challenges?
How do you communicate what a mathematical model is saying to a physician and their patient? We've started to poke around with this and work with close physicians about what they would want to see in a, quote, digital twin report. We have to mathematize oncology. It can't be this trial and error stuff for our kids. We have to get beyond this. I've definitely talked to clinicians who have said, yes, absolutely, this is phenomenal. And then I've talked to other physicians who will be like, whoa, slow down. I can't be changing therapy on the fly for each individual patient. We're kind of trying to go forward now is our mathematical model can then can identify a whole suite of interventions that would, in theory, outperform the standard of care the patient receives. Patient comes in, presents with disease X. There's typically 3 to 6 different options that they could receive that are approved for treating them. And so what we can do is we can do our modeling to figure out the best therapeutic regimen.
Because otherwise the physician could potentially leave treatment options on the table. So what about other oncology areas beyond breast cancer?
This generalizes to other solid tumors. So we're working mostly in breast cancer, brain cancer, and prostate cancer. The really nice thing about having a mechanism-based, a physics and biology-based model underneath your predictions is that the equations are, of course, agnostic to where you're applying them. I would like to contrast this with what is currently the shiny object in oncology right now, which are the methods of artificial intelligence and big data. Artificial intelligence requires you to have a very large population-based training set before you can make a prediction about an individual. You want to use any kind of AI engine to predict the best way to treat Miss Jane Doe, you have to have a training set of thousands of patients that are similar to Miss Jane Doe's subtype of breast cancer, and that training set has to have in it all the possible therapeutic regimens that she could receive. And that training set doesn't exist, and it will never exist because the diagnoses are getting more and more specific.
Now we've come back to discussing where the application of artificial intelligence is valuable and where it might not be.
Exactly. And of course, it's the choice of the data you use and how it depends on what you're trying to achieve. Maybe we use AI and big data to discover drugs and therapies, and maybe we use personalized data for more efficient patient treatment, especially in areas like oncology, where every cancer is different.
And remember, don't necessarily trust averages because they're not going to give you reliable data. There's that old cliche about averages being dangerous. And though we love it as a concept in statistics, they do say there are lies, big fat lies, and statistics. Let's wander off to school, Connor.
Let's do that. Let's do every day as a school day. What have you learned this week, Dodie?
Well, along the theme of fooling the big thinkers and the big data, I read a really funny story, again on science.org. Thank you, science.org, for always having something fun and weird and wonderful to read about. There were 2 researchers who made a fake academic profile for a character named Larry Richardson, pretending he was a mathematician and, citing all sorts of research that Larry Richardson did. Larry Richardson turned out to be their cat.
Okay, so the cat was doing research?
No, the cat was a fake profile, and these researchers were showing how easy it is to make somebody look famous in science with fake information. So, how do we decide who is important in science? How do we decide what to believe on the internet? It comes back to, ages ago, Connor, do you remember we did the episode about makeup companies that make claims about scientific? It's just verify.
So that's super interesting. I learned something along those lines, which is also very relevant to today's episode. I was reading a paper that explores the future of AI and focusing on something called model collapse. And this phenomenon has emerged as a really important concern. And it essentially says that as the average data out there starts to be data that has been generated by large language models, the next generation of generative models, such as ChatGPT 50 or what have you, is going to be basing its large language model on data that's been generated by large language models. And what happens, of course...
Oh, it's incestuous. The data is going to be born with a tail.
it goes round and round in circle. And these researchers have identified the fact that this predicts something called model collapse. And it's this degenerative effect where the AI starts losing touch with the true diversity of data. So you're training an AI exclusively on a diet of AI-generated content, and over time, it begins to forget the richness of outliers in data, and it focuses on a narrow, self-reinforcing view of reality. It builds its own echo chamber.
Right.
Essentially confused and useless. So we've got to really, really be thoughtful about
got to keep that AI fed with fresh meat.
That's exactly right. And when you have AI models that are trained on data that's been generated by AI models, you are not going to get a useful output. So there we go.
Okay.
Stay curious, folks.
Our producer is Beth Armit Brewster, editing, mixing, supervision by Banda Productions here in Sweden, music from Epidemic Sound. My name is Dodi Axelson.
And I'm Conor McKechnie. Make sure you rate us on Spotify or whichever platform you use. You will see us when we come back with another episode of Discovery Matters.